bioinformatics and next-generation sequencing informatics, and sort of what some clinical applications are and how you may be using some of these things in your practices in the future.
Okay, so I give this lecture to this sort of class a couple times a year, and I've tried today to strip out some of the more technical computer science parts and talk more about the actual application to patients. If you want to go into more detail, we can either have a little digression, or you could follow up with me, or take another course, or go online, that sort of thing. So this is going to be a pretty brief overview, and it's going to sound super easy, but there's a lot of detail behind. So the basic point here is that DNA sequencing has gotten dramatically cheaper and dramatically greater output in the last decade or so. It's a bigger jump in technology than any other that I can think of anywhere in science. I mean, it would be like, going from looking through a piece of glass to a radio telescope in 10 years. And as the sequencing gets cheaper, more scientists are using it for more research and publishing more papers, which is what this thing is showing. And in fact, some of the other hot technologies from the previous decade are showing that plateau effect, where it's not that people aren't doing research anymore, but it's not expanding rapidly and taking over new fields, while DNA sequencing is taking over new fields. People are shifting their investigations from other modalities to use sequencing as their primary data acquisition method. So we start with sequencing back in 1975, invented by Sanger, and this was the state of the art for about 20 or so years, where you ran 4 different reactions in 4 different tubes, for each one measuring one base, and then you merged those 4 tubes and you ran a big long gel, and then by hand you wrote on the side what letter you thought each band represented. And then somebody else did the same thing, and then you typed it into a computer and you saw if it meant something. And using this sort of technology, a really good grad student who was likely to get their PhD could get about 1,000 bases per week of data acquired. This was how I got my PhD, although this is not my gel. Around 1998, a little earlier, a company called Applied Biosystems improved the Sanger technology 2 basic ways. Number one, instead of using a single color radioactive label on the DNA fragments, they added 4 color labels so that all 4 bases could be determined in the same lane, which made it a little bit easier to sort them, because you couldn't get 2 right on top of each other the way you could here, where they were kind of hard to tell apart. And also that it could be read automatically with a fluorescent detector hooked up to a computer. So instead of squinting at a piece of X-ray film, you had a printout at the end of the day. So it increased the throughput, maybe 1,000-fold, maybe 10,000-fold. And this is how the Human Genome Project got completed around year 2000, 2002, depending on when you put the benchmark for completion. And the way they scaled up the Genome Project was to get more and more of these ABI machines in bigger and bigger rooms. So this was not actually an inflection point in the technology as much as you might think. I mean, yes?
Was the government the first to complete the sequencing? Or?
Depends on how you look at it, but yes.
Okay. The whole Craig Venter.
The Venter, well, they used the government data, added some more, claimed ownership. At the last minute, they all shook hands and had a big hug. So it was a joint project. Certainly they contributed. They contributed more in the area of what they call shotgun sequencing. Rather than breaking things out by one big piece and then breaking that into littler pieces and sending those littler pieces to different labs and then bringing the data back, which was sort of a step-by-step approach. They said you could shatter the whole genome into many tiny pieces and then reassemble it computationally, which was, in fact, the way of the future. But at the time, they used the step-by-step landmarks to build their assembly, so it couldn't have been done without what had been done before. They jumped ahead a year and a half over what the original government plan was, so the whole thing got done faster because of their contribution.
And whose genome was it that was sequenced?
Yeah, that's a big secret.
Oh, really?
The official rule is that it was a composite, because they had some of the sequencing was done on these weird hybridomas where you put one human chromosome into a mouse cell, and so each chromosome came from a separate event in a separate lab. And then there was obviously some of Craig Ventnor's DNA in there as well.
So if you actually tried to, say we had technology to actually produce, people, is it viable? Is that a viable DNA sequence?
Oh yeah, it's a viable sequence. It has correct sequences for all the essential genes, more or less. I mean, it's a consensus, so it has more common alleles than any one person would likely have. You know, it's like if you had somebody who had the most common allele at every position,
So
something
it's
like
a perfect
that.
average
It's not
person.
perfect, it's just, it has common
perfect
alleles
human.
at a lot of positions where an average person has a certain number of variants, which I'm about to get into. Also, you can take the Ventnor sequence was done separately with another technology a few years later, so you could compare his to the consensus and see where there's similarities, how much of his might be in there. So the take home from the first few slides, DNA sequencing is improving at a phenomenal rate, and it hasn't really plateaued. We hit these little mini plateaus for a year or so, and then it breaks through again. So I don't think we've hit the limits of what the current technology can do. It kind of like Intel chips keep getting more circuits per square inch or per square millimeter. This DNA sequencing is on the same kind of relatively rapid increase rate, but they're in fact increasing more rapidly than computer processors. So this is probably still true. The output of the machines is doubling and the cost is falling by half in each of the past 5 years. So very rapid exponential growth, and it's faster than the improvement in computers, which actually means that it requires more cost in computers to analyze the output of the machine every year. So the relative budgets of the bioinformatics group has to go up. And when you get such huge change in net output, you get qualitative changes as well as quantitative changes. New things that you hadn't thought of before become possible. And I'm going to try and dwell on some of that. So the current technology, as of maybe last year, because they're always out with a new model, but we have one of these still, the Illumina HiSeq 2500, produces billions of reads per sample, 450 billion base pairs of data. And that's actually more than you need to sequence an individual human genome. So they're coming in on the $1,000 genome, depending on how you slice it. Some machines can produce human genome sequence of clinical quality for somewhere near $1,000. That's not counting the cost of acquiring the sample or analyzing the data. Illumina is far, far out in front of any other companies, so I'm going to focus almost entirely on Illumina data. But leads in this kind of technology are relatively ephemeral, and someone can come up with a new technology and kick them off in a relatively few years. At the moment, there's not an obvious challenger. So our sequencing lab is now all Illumina machines here at NYU Medical Center.
So when you say 1.5 billion reads per sample, does that mean there's some duplicative samples because you're looking at so many different cells at once? Or?
Well, a sample in this case means a library, a tube of DNA that you've ground up and attached primers to. And there are ways to put multiple samples into the same lane and then sort them out bioinformatically at the end. But per sample is an Illumina term that they mean a lane of their machine, which is vaguely equivalent to a lane in the ABI machine, but billions of times more data dense. And I'm going to actually show you some pictures. I did keep a picture. So this is how the Illumina works, very, very briefly. You start with your target data, target DNA, which could typically be a human genome or a research animal. You could also be doing a more reduced version of data analysis where it starts with PCR and you're focused on only certain portions of DNA. Oh, well, that's kind of a bummer. Can we get the...
I think if you, yeah, it's just if you,
Oh, just went to sleep.
yeah.
Okay, fine. So you chop up your target DNA into smallish pieces. Currently, we're using pieces that are maybe 500 bases long, but that changes with the technology, and there's even different approaches to the same technology that might use longer or shorter pieces. You attach on some primers with specific sequences that are used to attach the fragments onto the flow cell, the piece that goes into the sequencing machine. Inside the flow cell, each individual cloned fragment is amplified by a modified PCR into a little cluster. This is a signal amplification step, and simply to allow enough fluorescence for the machine to detect. And this is an area where they can obviously improve. They can make the clusters smaller and their sensors more sensitive. They can smush them into smaller space. And this is constantly happening. Then the Illumina chemistry kicks in after you've built these clusters, and it sequences all of the genome fragments simultaneously, all over the plate, and it uses image analysis technology to sort them out. So you have billions of tiny glowing spots, and you add one DNA letter at a time. So you have a cycle of A, and all of the fragments where A is the next letter ready to be sequenced will glow. And then you add a G, a T, and a C, and therefore every fragment should light up with one and only one of those reagents. And then you decouple, meaning you remove a terminator, and then you go with another set of 4 reagents, G, A, T, C, and again, every spot should glow only one time. You take all that image data, process it with a lot of computer technology, and you record a sequence of letters for each spot on the flow cell. It's a giant grid with billions of little locations.
And each spot is about 200 base pairs?
Well, it depends on how many cycles of reagents you use. The way that we're doing it most commonly right now is we're doing 100 cycles. So 100 letters are recorded, and then we turn around and we read 100 letters off of the other end of the same fragment. It's called paired end reads, and those turn out to have advantageous properties for the bioinformatic analysis. So if you can get 200 bases, it's somewhat better to get 2 reads of 200 bases that are separated by about 300 in the middle than it is to read 200 straight through. I'll show you a little bit of that, but I'm not going to dwell on it. But that's what Illumina can do. There are some versions of the Illumina machine that could read as many as 250, maybe 300 bases in a row, but nothing up to like 1,000, which is what we were able to get from the older Sanger technology. There are other companies that are making other machines that can read much, much more data linearly in a row, but much less total data out of one run of the machine, and therefore much less total data per dollar, which is where the market is focused right now. How can we get complete coverage of the human genome for a reasonable price? And good accuracy. The other machines also have accuracy problems. Illumina has really focused a lot on data quantity and data accuracy. And they're the leaders right now. I won't say it can't be better, but they're the leaders in the market right now.
For the amplification amplification phase, sorry to go back for a second, is there any risk of bias being introduced by selective amplification, or?
Doesn't seem to be. I mean, there's been a lot of bioinformatics studies saying, how good are we covering the genome? How random is it? And the problems don't seem to be at this phase. The biggest problem is in the phase where we chop, because not all base pairs break at the exact same efficiency. There's proteins associated with the DNA. There's actually bias in the sequence itself, how tight it holds together under various shearing. So you never get a pure coverage, but we usually model it with a Poisson distribution, you know, like bombing London. It's kind of random. In some places get hit more times, and some places are low. And there is bias there. It's not purely random, but it's a model. This is like zooming in on the clusters and zooming in on the fluorescent detection. I don't think I need to go over it again.
And this can come from any DNA, like any way it was obtained?
Yes. And that's actually part of the attraction of this system, is that you can use the exact same system to sequence microbes in infectious disease or virus or something else.
But like, what is your input into the actual processing system? Like, let's say you have like saliva or blood or tissue.
Right, so your input is a tube of DNA, whatever the investigator wants to hand
So
off
it
to the
doesn't
lab.
matter.
It doesn't matter. It needs to be reasonably pure and to come from the organism you say it comes from, and that's about it.
So how much preparation does it take to get to that state?
To make the DNA?
No, no, no.
To go from the DNA
to
to
go
the...
from like a blood sample to, you know, a vial full of DNA.
Like if I want to get.
Just run it over a Creogen column and lyse it, 10 minutes in the lab.
Okay, so it doesn't take that long.
You clean up and then you lyse it, you add alcohol.
But like
and
blood, you have to run a column. You can't just like.
Depends.
spin it
I
off
mean,
and
most
give you some
people run a
serum.
column because it's easier than adding a bunch of different reagents and going through a bunch of centrifugation steps. I mean, you can make DNA from anything, but different organisms have different, contaminants. Plants have starches. It's a pain in the butt. Human cells are actually really easy. Blood cells are like one of the cleanest sources of DNA, so you can use less.
How much blood do you use?
I don't know, tens of nanograms of DNA. I don't know how much, certainly, a typical blood draw is much more than you need.
Got it.
I'm not going to focus on this too much, but this is what we get out of the machine. And so if you deliver a sample to the sequencing lab and you have no bioinformatics collaborators, this is what you get back in the mail, except it's maybe a terabyte, like a 1 billion line file. Each one of these lines is a sequence of one spot. It also has a quality line, which is in ASCII. And so obviously there's software that can read this and take it to the next steps. And typically a clinical investigator would work with a bioinformatics consultant on the data processing. Your goal is generally, but not always, we'll talk about this, to take this very, very giant data file of raw data and break it down to a list of variants, actual mutations, differences in the DNA of your sample compared to some reference, such as that reference human genome that was determined by the Human Genome Project. And this is only a moderately sized file. The typical human differs from the reference genome in maybe only a couple million places. So this is a megabyte file rather than a terabyte. You could easily walk around with this on a flash drive. Although it would be a lot to read through 1 million variants and try and figure out which one is the disease-causing variant for your patient. So again, software is going to be needed here.
So that's all the exons and introns?
Depends on what you've done for your sequencing, right? This is just the data output here. And if you had, instead of shearing the DNA, had gone through a step of specifically PCRing the exons or target genes or you had a panel, then that's what your data would reflect, more or less. This is just data. After mapping, then you would know more. I'm going to get to that in a second. So the NGS data output has some characteristics. The coverage is uneven. There's some bias. So we could model this as a Poisson distribution, which means that there's going to be some spots on the genome that have lesser coverage. And when we're trying to find mutations, there may be a limit on where we have enough accuracy to say, yes, we know that there's a mutation here. There could be a spot where we say, oh, we can't call any variants at that spot because the coverage is too low. There's also can be places where the coverage is too high because the sequence is repeated a lot, and therefore you can't necessarily map the variant to one unique location on the genome. The machine itself has errors. It's less than 1%. It's probably more like 0.5% on average. But again, there's bias. There's places where more errors tend to occur. That's what I said, systematic. And some of those systematic biases are actually caused not by the machine, but by the software that we use to process the data. There's places where the software has a hard time figuring out where exactly a given chunk of sequence fits on the reference genome. And that can create false sequencing errors. And then there's this quality string that goes with every read. You want to avoid calling a mutation from a low quality read that the machine itself flags as possibly not correct. So what are the applications for clinical medicine? Cancer has been the clear leader here in terms of attracting attention. You can identify genetic characteristics of a tumor. Every tumor has a different collection of mutations because it's evolved according to a somewhat random process. There also might be a possibility to match a patient to a drug. If you have this mutation, then this drug will be good for you. If you have this other mutation, this drug will not be good for you. And those could be either drug targets, or they could be drug inactivating or drug resistance mutations. There's also the possibility of diagnostics here, where a given mutation predicts a highly aggressive tumor or a less aggressive tumor, or the response to treatment. This person is going to be a good responder or a poor responder. Another area of medicine is genetic disease, particularly diseases that are not diagnosed by the standard panel of genetic markers, which is sort of a combination of protein, metabolome, and some DNA sequence markers. So if a child is exhibiting symptoms that are reminiscent of a heritable disease, but they're negative for the standard tests, then we start sequencing the genome. And that can be a grab bag, but sometimes you can identify a new mutation that is strongly suggestive of the symptoms that the patient has, which then suggests a particular therapy that might be appropriate. Pharmacogenomics has to do with any other sort of disease. There are mutations for drug metabolism, there are mutations in drug targets, and this is the whole field of personalized medicine. There's the area of infectious disease where mostly we're sequencing the pathogens, and we could be doing this in a outbreak situation where there's a new pathogen emerging and we need to get genetic information very quickly. We could be doing surveillance on a large sort of population base. What's the rise of various genes and resistances in the whole population of pathogens? We could be mapping novel drug resistance mutations that are emerging in pathogen populations. And then this is not so much a clinical approach, but it could be, is measuring gene expression by using sequencing, by saying, okay, this gene expression profile is, say, diagnostic for more aggressive cancer, or diagnostic for good response to this particular drug. Rather than looking at the individual mutation, now you're looking at it from a more functional standpoint but again, using sequencing as your data collection method, just because sequencing is fast and accurate.
May I ask regarding the RNA sequencing? It's something I've been trying to learn in the last couple of months, and I just wanted to ask, you're making these cDNA libraries, these clones, and is the purpose of that really just to use the same approach?
Yes.
Just with DNA as your starting material?
Yes.
Okay.
And there's some advantages of using sequencing as opposed to other methods for measuring the levels of RNA in a cell population. And I'm not going to give that lecture right now because I don't have enough time, but that's clearly, lecture 3 in the process. All right, so we're mostly going to focus on finding variants because that's the most, the area that clinicians talk about the most, we'll put it that way. There's a lot of human genome sequencing. Probably Illumina machines are used to sequence human genomes more than they're used for any other single application, and most of that is to find variants. And some of that is pure population sequencing, 1000 Genome Project. A lot of it is cancer sequencing, either research-based, we're accumulating information about a particular cancer type and trying to learn what genes get mutated in this type of cancer, or we're applying that data and we're sequencing the genome of an individual patient, comparing it to what we know about cancer, and trying to make some sort of diagnostic decision that's going to benefit that patient, that yes, you have this mutation and therefore this drug is going to be good for you. Right, so I already talked about how sequencing is not perfect. The data output has some issues, and so we have to try and overcome that in order to get a clinical quality set of variants for this patient. This is what the analyzed data might look like in sort of an intermediate level, where we've done the sequencing, we've mapped the individual reads back onto the genome, and we've highlighted all the differences. And what we see is often like this, where there's a lot of differences that are sort of scattered about, and we consider those mostly to be just sequencing errors. And then here's a stack of variants that looks pretty suggestive to me of a heterozygote, right? About half of these positions are C, and and a little more than half are A. A is the reference letter, C is a variant letter. This could be a mutation, or it could just be a common allele. We might see C very commonly when we look at a database of sequences taken from a lot of people. In order to interpret a thing like this, we need a lot of reference genomes, like the 1000 Genomes Project, to say, this is a known variant, it's been mapped in our database of known human variants, it has either some clinical phenotype significance or more commonly no clinical phenotype significance, we can pass on.
And usually how many, like if you're looking at like a rare genetic disorder, like how, like, because obviously you need a lot of references compared to, but how many of the disorder do you need in your like comparator arm?
There's not a protocol for that.
But
I mean,
it's
people
usually
have
more than
done,
one, right? Like, it's
yeah,
usually
but
like a
people have
series.
had success with N of 1, or sometimes they have like 10 in their trial, and they find 10 different mutations, one for each person, that have nothing in common.
What is the like main approach usually, to run all of them singly and then together, or usually you run like the group of people with the condition together first, and then if that doesn't pan out,
Well,
look at each person?
ideally you need a big batch of sequences, because your data collection and sequencing has a little bit of bias to it. So ideally it's nice to run a few hundred genomes all in a batch, and then the ones that have specific variants that stand out will be easier to map. If you have just one, then you have to use somebody else's batch of genomes as a comparator, and it's not quite as good. So I wouldn't say there's a protocol for this. I mean, people have their study design based on factors other than how best the sequencing machines work. There's all the protocol has whatever characteristics it has. This is how many patients we're able to acquire. This is the amount of money we have. So we have to sort of deal with that. But it's nicer to have a bigger batch so that the error sort of averages out and the real variants rise to the surface. Here's a really obvious, looks like it's a pair of mutations. You might call this a haplotype. It's different than the reference. Also, it's present on many different reads that begin and end at different places. So these are clearly variants, but again, we would need a database to tell us whether these are just ordinary common variants that everybody has, or that many people have, or whether this is a unique pathology related to this individual, or a unique disease-causing variant that we've seen before. That would be even better. Heterozygosity is really common. Humans are diploids. So a variant frequency of 50% is a successful run, but obviously what do you do with a variant frequency of 20%? That's probably also a heterozygote that you've just mapped by chance at a lower frequency. And then, well, what do you do with a variant frequency of 5%? That's not in our model, unless it's a cancer genome and it is itself, the cells differ, and it's a relatively rare mutation in the cell population that you've sequenced. Okay, so I talked about annotation now. Each one of these variants has a position on the genome. We know whether it's inside the coding sequence of a gene, in an intron, somewhat upstream, or completely far away from any gene, intergenic. If it's in a gene, we know whether this gene has been implicated in disease before. We have a database of human genetic diseases called OMIM. We can say whether the change we observe is a coding mutation, meaning it changes the amino acid, or non-coding. So synonymous, non-synonymous, changes, doesn't change the amino acid. It could affect splicing. We can attempt to predict the effect of an amino acid change on the protein structure and function. That's a little fuzzier, but people have done it. If it's not in the coding sequence, but it is in a promoter sequence, we could attempt to predict an effect on gene expression. That's even much more fuzzier. And then we have this problem. What's the penetrance of this variant? Do we know whether having this variant in a heterozygous form has any known effect on patient health? What if it's only statistical, right? It increases your likelihood of Alzheimer's by 5%. Is that a reportable discovery? A lot of the database is in that form. Very weak associations between a variant and a phenotype.
It's all correlative too, right? It's all correlative. Like you don't necessarily know that there's a causal, it's in the causal pathway.
No, causal, it's associative, right?
Yeah.
More people with disease have this variant than don't.
Right.
That's the best we can do right now, most of the time. Very rarely we see a variant that, mangles the protein and it's, it's sickle cell. But those were the easy ones, and mostly those have already been nailed, and it's the fuzzier ones that we're struggling with now. So each genome has about 2 million variants compared to the reference genome. Everybody.
Are the reference genomes, are any of them public?
They're all public.
They're all public, and they're all they're all from the same. Like
well,
what?
the reference genome,
Like, do you have to
the
adjust?
reference genome, GCRC38, is a collaboration of the whole world. And it tries to be as correct as possible. I don't know how you deal with correctness. Then there's another class of genomes, the 1,000 genomes. That was 1,000 volunteers, research volunteers. I wish I knew that.
We have, 20,000 genes?
Well, there's a 10K that's been published in the United Kingdom, the UK 10K. So that's the largest number that we have now. But the 1,000 genomes is easier to work with, and it's been processed to a higher level of analysis. So the annotations on those variants are easier.
No, but I mean, in our genome, we have 20,000 genes, or
20,000 genes, yeah. The 2 million variants are randomly distributed. Most of them are not encoding genes. In fact, they're selected against coding regions. They're more prevalent in the non-coding regions. Only 1% of those 2 million are in coding regions. And of that 1%, maybe half are non-synonymous, changing the amino acid. And of those, it's very hard to say what the penetrance is, what the effect on the protein might be. It could be subtle. So cancer genomes have more mutations because one of the steps in progression to oncogenesis is failure of DNA repair. And different types of cancer have different levels of mutation. Some seem to be pretty focused on certain target areas. Others have this hypermutator phenotype where there's a huge amount of background mutation. And so we have this problem of cancer-causing variants versus the passengers. I don't quite get why this is doing this. So the passenger mutations are just there because the cancer genome is not correcting its DNA, whereas the oncogenic ones are actually driving the cancer. They're leading to higher levels of replication, more vascularization, et cetera, et cetera. And sorting those things out is a current challenge. And part of that challenge could be described as how to focus on the relevant data. Do we only look at protein coding mutations, or are the mutations that might regulate gene expression also relevant? So there's too much data right now. So if you give a clinician a report with 1 million variants, they're not going to be super happy because they're not going to be able to make actionable decisions very quickly on that report. We could classify the diseases as being simple or complex. Simple diseases have a single cause. A single gene is damaged, which leads to an observable phenotype. We have a lot of models for this: cystic fibrosis, muscular dystrophy, Tay-Sachs. Then we have complex, where there are many genes that contribute either in a single individual or different people have different mutations that lead to a similar phenotype. And so autism, schizophrenia, Alzheimer's, asthma, heart disease. So these are things where we talk about risk and accumulating risk factors, but that language is pretty fuzzy. And it's difficult to give a patient a report saying you have the following risk factors, but we don't know how they interact. One might trump the others, and that's the only one you need to worry about. These two may interact synergistically, and therefore you have a much higher risk. Or this other one that we don't even know about trumps all the others and suppresses them, and you actually have a much lower risk. So that's kind of where we are now. So we have the sequence data, which is pretty accurate. We can annotate. We have some databases, but we don't know how to integrate all the data very effectively unless we have a very tight diagnosis that we're looking at. Is this gene affected? We could answer that question, but we could have answered that question 20 years ago without the next-gen sequencing. The whole genome gives us a lot of data, and it's confusing. So we go from a single base diagnosis like sickle cell, single gene like muscular dystrophy. You have to sequence the whole thing because there's many different mutations in the population. You could sequence a panel. That's where we are now with cancer and clinicians, and I'll talk about that a bit more. This is a bunch of genes that we know are important. It's only 100 genes, so we could focus our attention on just 100 genes. And we could tell you whether you have or do not have any mutations in those 100 important genes. We can't tell you what's going on in the rest of your genome. Then there's something called exome sequencing, which is focusing on the coding regions of all the genes and ignoring the other 98% of the genome that has mutations that we don't know the functions of. And this is very popular right now in diagnostic sequencing, where the focus on the single gene has failed, so now we want to look at the whole exome. It's also very popular in a research mode where we're trying to collect as much information as we can to associate disease with genotype. And then there's the whole genome data, 3 billion bases, 2 million mutations per patient. All right, I'm going to skip over translocations and gene fusions and copy number variations because those are other modalities of genetic change that are different than the single mutations. And I don't quite know how to integrate that right this minute, but the FISH is another example of an assay where you could look at translocations, a chromosome breaking and rejoining. You could assay that by sequencing, but it's a little bit of an inefficient means of doing that. And it's not the ideal technology, but definitely people are working on that. So the biggest challenge is interpretation, right? We have the ability to gather this data pretty efficiently, and it's reasonably accurate, but we don't know exactly what to tell the physician or the patient about their genome. In cancer, we now have some target genes that are bad actors, and we could look at those genes pretty carefully, but we don't know about the rest of the genome. We need a little more data. It's in a case of where a child has a syndrome that looks like a genetic diagnosis. Again, we know some genes we might want to look at. We might know other genes that have a red flag. Here's a structural mutation in a gene that we didn't think about being associated with this disease, but maybe we could sort of alter our model a little and say, oh, maybe this gene is important for this disease. And then there's the healthy person problem. You have lots of mutations in a healthy person. How do we interpret them? It turns out that everybody has several serious structural mutations that break the function of an important gene, yet they appear to be healthy. So we're in this state right now where we're trying to collect large data sets to associate specific mutations with specific phenotypes. And this is a sort of a high priority research area for NIH, et cetera. Clinical database of mutations. And then we have to worry about how do we deliver this information in a CLIA certified, accurate way. So there's technical validity, right? Was the assay done correctly? Is the sequence mutation ascertained with high certainty? And then there's the predictive power. What do we know that this mutation does for your health? And that's the fuzzier place. Right, and so this is just a sort of a case study example. If you sequence a known disease gene with a family history of a specific genetic disorder, you're looking for, is the family mutation present or absent? So this is like a 20 years ago approach versus sequencing the whole genome of a newborn, looking for anything that might be predictive of disease. Just can't be done right now. We don't know enough. We know what this is and we know how to treat it. That would be clinical validity, right? Or utility. Will this result lead to an improved outcome for this patient? And in this world of too much information, is it better to focus only on actionable tests? So here's an example of a group of children early on in the sequencing era where they were having this congenital chloride diarrhea. So they had genetic evidence. The molecular diagnosis was based on finding a homozygous missense mutation in this gene. And then after they figured this out, they were able to find 5 additional patients in the world who had the same or similar mutation in this same gene. So this is the example of adding another gene to the catalog of known genetic diseases, done by looking at a child with an undiagnosed suspicious disorder. Here's another example. There were family-based exome sequencing in 10 individuals with unexplained mental retardation. So 10 different subjects from 10 different families, and they found 9 genes in the 10 individuals. So each one had a different mutation that led to their unique, similar seeming mental retardation. So this is a case where we're not converging, we're expanding. It seems like, not that every mental retardation is unique, but there are many, many, many causes for what could be diagnosed similarly. And then here's an example from TCGA, this NIH-sponsored cancer genome study. Exome sequencing uncovered a median of 16 mutations per tumor. 8 of these genes contain mutations in more than 3% of the samples. So that means that a lot of those mutations were not common. Half of the frequently mutated genes fell into certain pathways: histone modification and chromatin remodeling. Other genes that were mutated above background frequency were in these other pathways: Wnt, signaling, hedgehog, etc. So this is the kind of data that we're accumulating, but it's still coming a bit slowly. So here's a private company that decided to take what we know and focus on just what we know about. So they built a panel, 182 genes for solid tumors. They built a second panel for blood-based tumors, which only overlaps partially. Different mutations are more common in leukemias than they are in solid tumors. And they started sequencing a lot of patients, and in the last year they've been certified in a number of states, and they're starting to get Medicare reimbursement. Some hospitals have partnered with them, so they send all their samples to Foundation Medicine. Other hospitals have decided to make their own cancer panel. The advantage these guys have is they've sequenced that same panel tens of thousands of times now, and they can call mutations in that set of genes with great accuracy. They know all the biases in their system, and their mutation calling is very accurate. And they're starting to get 5-year follow-up data on patients. So now they can start to associate a mutation of unknown function with drug response, with how aggressive that tumor was in that patient. It's still a bit of a data juggle, because in those 100 genes you can obviously get quite a lot of different mutations. So associating a variant with an outcome still challenging, but they're accumulating data more rapidly than anyone else on the genes that they're targeting. Illumina has their own panel. A bunch of other hospitals have developed their own panels. This is currently the most popular approach in cancer genomics, is focusing on where the light shines, where we have some information about genes that are known to contribute to cancer and may be potential drug targets.
So is this achieved at the amplification stage, because these are the targets I want to amplify more?
Actually, there's a way of selecting. So you cleave the genome, and then you have a hybridization target, a bait they call it. And so you wash the fragments over the targets. The ones you want stick, and then you wash everything else away. You release the fragments and put those into the sequencer. So it's about 95% efficient. So you have a little background, but most of your sequencing is focused on the genes of interest. In addition, because you're doing it by hybridization of randomly cleaved fragments, they stick out a bit beyond the target region. So you get the coding sequence plus a little bit of flank with kind of a normally distributed tail. So that's what the data looks like when you do exomes. I mean, this isn't exomes, this is selective for just 100 genes or 180 genes. The same approach would work to exomes. You'll be selecting the coding sequence of every gene. And really the data processing and the lab processing is really the same. The advantage of the panel is you can ignore more of the data and not worry about it, and focus on the stuff where there's some likelihood of predicting what value it has for the patient.
Is this FDA? You have to go through FDA approval?
No, it's apparently not. It's a diagnostic. Well, I guess if it's going to be done by Medicare, yes, because they're approving the accuracy.
Right.
And you are although the company isn't giving you any health advice, your oncologist would be reading that data, reading the report, and making the advice.
Right.
So all they're saying is we have a known mutation in this gene.
Right.
So I think it's mostly about clinical accuracy here, or
Yeah.
technical accuracy.
Right. Can't imagine each panel trying to get FDA approval.
Yeah, well, if it's a lab-based test, right? So they have provisional approval.
Right.
If you're Mount Sinai or, you know, MSK, you can say, okay, we have an internal lab-based assay, and we validated it in this way.
Right.
And we're not doing anything other than using it to advise our own oncologists. It's not open to the public. You can't send your data, your cancer genome, and have it run through a panel. You have to have it sent by your oncologist. If it was open to the public, they'd have a lot more approvals they would need. All right, I'm out of like 3 more minutes. So these are thought questions for you guys. Might work as an exam or a quick quiz. So obviously I've presented big information versus targeted information. And the downside of the big information is we don't know how to interpret it. The downside of the targeted information is the false negative, right? We have no observable mutations in any of our 100 target genes. Doesn't mean you're healthy. It just means you might have mutations in places where we didn't look. You might have mutations that affect gene expression. You might have a lot of other things going on in your genome. It's just that we don't know how to interpret those things as well. And then there's this idea that genetic tests produce unintended results. So we were going to be looking at this, but in fact, we found this other thing. You have a mutation that gives you a high risk of Alzheimer's or something like that. I have a few more sort of ethics-related slides we can go through in just a couple minutes, and maybe I'll leave the slide deck with Yin so you could look at it some more.
Has anyone used any panels here, or ever, or seen people? Do you guys use it?
We use a ton, yeah. Well, NYU is 3 for like BRC mutations.
Uh-huh.
And a bunch of.
So those are local panels, not a company providing it?
No, it's Ambry. One of the big ones we use is Ambry.
So that's a company-based panel.
And that's the patient themselves provides like a blood sample, or it's like
Right.
the tumor is sequenced, like the tumor is.
A blood sample.
Oh. Yeah, we use
So
it as well.
you're looking
in,
at genetic risk factors, not actual tumor-based mutations.
Well, now we're going to be looking at tumor-based. We're taking tissue samples and looking back at.
all our
Okay.
brain tumor patients, intraaxial tumors, I don't know if we're still doing it, but they get a blood, we draw a blood sample of theirs and test it for 1p19q to predict aggressiveness of the tumor.
But that's again in the patient's germline mutation.
Yes, yeah, germline.
So that's sort of a susceptibility gene.
Yeah.
Now, how do you counseling? Are there specific protocols? So if someone is positive, so then you just go down this route?
I think
or like the counselor shows up, or is there like the specific actions associated with?
well, I mean, so after their surgery, they typically have to meet with their neuro-oncologist for, whatever treatment they're going to undergo afterwards, whether it's chemotherapy, radiation. There's usually standard protocols, but I think now based on certain mutations, there's certain clinical trials you can be enrolled in.
Got it.
And it predicts response to certain chemo agents and affects median survival.
Right.
Like that.
But it's all at the trial phase. It's not necessarily like evidence-based practice. It's not in the.
No, we can't. I don't think we can unequivocally say if you have this genome or if you have this mutation or not, that this will 100% predict and this is what should happen. It's
Right.
still like we're just assessing, figuring it out.
Yeah.
So this is like a summary of ethical considerations for genomic testing, which is really no different than ethical considerations for classical genetic testing going back 20, 30 years. So there's the possibility of genetic discrimination. There needs to be informed consent before you begin the process, which is hard because it's hard to inform people about all the possible outcomes. There's privacy and confidentiality of the data, and there's been some horrible fails in that area recently. Diagnostics, predictive testing, which has variable predictive value, how to report the results back to participants, how about other family members, do they need to know, what if they don't want to know, if they say have a breast cancer susceptibility gene, that sort of thing. And then what about, this is bizarre, but government and law enforcement can get access to genetic information. And this has happened with some epic fails in the recent past. So someone submits to a genetic test for some medical, or they're interested in genealogy, and their data goes on file, and then it's subpoenaed by law enforcement.
Did that happen?
Oh, yeah.
Was there in like a sentinel case?
Yeah,
That's right.
I don't think I have it in
Going
this
after
slide
it.
deck actually, but there was this crazy story. Yeah, I only put the good ones here. So there was this person who participated in a Mormon database where they were trying to link up people because that's an important Mormon belief. So he sent his DNA to this company that was endorsed by the Mormon Church. The company went bankrupt, sold their data to another company, one of these online genetic genealogy websites, and then in Idaho they were searching for a murderer, and somehow they got a judge to give them a subpoena on all genetic data everywhere in America. And they could subpoena from everybody all their genetic information.
And they found the killer
No.
in the database?
No, but they found
Like-
they found this guy
He was
though.
there.
They found this guy, but he was in Florida and the murder was in Idaho, but his son was in Nevada. And so they arrested his son.
That's messed up.
Well, you can. I think. I don't know if we're there yet, but there's.
But his
there's-
son
But he
didn't do it.
You can
How
take
do you know?
genetic data.
Because they had a murder investigation and he was acquitted. But they came to his door
doesn't mean he
because
didn't
his
do it.
dad, well, it does mean he didn't do it. They investigated him and somebody else was the one who did it. He wasn't in the state at the time. He couldn't possibly have done it.
That is crazy.
And he only had like a 25% relationship with the
Oh, okay,
actual
right.
sample.
Yeah,
The son's got a kid, and
That kid, right. But the idea is that any data is infinitely searchable and can be associated by people with high levels of ignorance with whatever they want. That was hilarious. I didn't give you that one because I didn't want to go that way, but yeah. Let's do a good
So
one.
let's see the good one.
Let's see a good one. So there's a really common genetic test going on right now, which is basically for Down syndrome. It looks at fetal DNA in the mother's blood. And I didn't think this was going to work 5 years ago, but it works incredibly well. There's been 800,000 women in the past year who've had this test. It's the most successful genetic test ever, economically, money. And what they're looking for is they have a high ability to diagnose Down syndrome early without, non-invasively, right? You don't have to go sampling cells from the fetus. There's enough fetal DNA in the mother's blood to unequivocally diagnose Downs. But in this case, they found in the mother's DNA a breast cancer mutation.
Do you guys do that downstairs?
So wait, what's this
Standard of care?
800,000
Yeah.
women?
Yeah.
What's
From
what's
maternity
NIPT?
Non-invasive prenatal testing.
Oh, okay.
It's mother's blood testing
Right.
that reveals mom has cancer. So
Right.
what happens is mostly you get mom's DNA, right?
Yeah,
Only
yeah,
like
right.
0.05% of the DNA is the child. But Down syndrome is really obvious.
Yeah.
And there's enough data to diagnose for sure. But at the same time, there's mom's DNA, huge red flag. She has this mutation that is suggestive of cancer.
So is this like DS frequently do this test?
Mhm.
Oh really?
It used to be reserved for only women who are like advanced maternal age or
Right.
had other predisposition, but now
It's becoming standard.
it's
of care.
becoming
Oh,
standard.
oh, that's interesting.
Does it only do Downs or is it?
No,
Breast
the
gender.
So you can
Yeah,
do
gender,
this
other
yes,
chromosomal abnormalities,
Yes, it is
but
not-
it's big stuff. It's not little stuff that they're looking for with the prenatal testing.
So this was a proteomic test.
Okay.
But yeah, this is just a
So it's like a
one.
Because David claimed
But
that
it's
this was a
very
proteomic
high.
test, not a genomic test.
Well, this was a
But
genetic
that part
test.
is,
So
yeah.
you will do this instead
Yeah.
of having CVS.
So does the company have an obligation, like do they
Technically, it's not
investigate
used
this
as a replacement,
active
but it's
proactively
like
or?
the way
Well,
that
in
it's
this
done.
case, they are in fact flagging these things. They have put up some software flags in their system, and they have arbitrarily decided to release certain classes of information back to the patient, and probably they have to include that in the consent when the person sends this in, that we are going to recontact you if we find any of these other sort of unintended findings, because it's a big problem to give people information they don't want. But in this case, he says we see about 40 a year out of 800,000 samples where it was necessary to recontact the mother, suggestive of cancer of the mother. And 26 out of the 40, so way more accurate than any other test I've ever heard of, are actually have cancer. So obviously they're only flagging things that they're really pretty sure of.
I think they were all early stage too. I think these were all early stage.
Yes.
Very
I mean, if
early.
you read the article in the Times, it was, caught early and she's fine now, and there she is with her baby.
How does the report show up? Are they just PDFs?
I don't know that.
It
Oh,
looks
she-
PDF. It's just like a.
It's like a risk assessment. Oh, no, it shows you like,
Yeah, it's a
Yeah.
PDF.
Yeah,
a
it
PDF
is-
that they
yeah.
upload, yeah.
And then
they
it's
send.
some.
And it categorizes like the different. It does like cystic fibrosis, the other trisomies. It tells you gender.
Yeah. And is it like yes, no, or it's like a risk number?
It's a risk number.
Okay. So even the gender is a number too.
No, they're usually pretty, like if there's no male
It's going to be hard.
DNA
They
in the
look
woman's
for, like,
blood, then
SRY. Yeah.
Okay, yeah.
So.
Right. But then the other things, they're all risk numbers.
So here's a fail, the opposite situation. I will get that back
The
somehow.
the Scripps company is the only one that does it right now.
They have some kind of a patent on this.
Oh, okay.
All right, so about 10 years ago, so earlier in the history of NGS, an infant with seizures was genetically tested by sequencing. This was not a whole genome or exome, so presumably they were sequencing a panel of genes that they were suspicious of, and they had an inconclusive result. You'll hear this a lot if you get into this business. A variant of unknown significance, a VUS, found in a potentially relevant gene. The doctor did not act on the inconclusive genetic results, including did not release that result with the family, and the child died. Since that time, this variant has been reclassified as a disease-associated mutation. So the question is, what weakness in the system can be improved? Clearly, this doctor did nothing wrong, because every assay has variants of unknown significance, and generally the protocol is not to burden the patient with, we don't know, here's some stuff you should worry about that we don't know what it means. That's not usually what they do. But maybe they should have updated the database and recontacted the patient or family when new information became available. Maybe the inconclusive results should have been shared in some form so they could go for another opinion. Genetic data sharing among testing labs. So this is just an open question. I mean, nobody's at fault here, but this is a case where data could have helped because they were treating this child incorrectly based on sort of clinical guidelines rather than knowing that this particular gene was mutated and therefore would merit a completely different treatment.
I guess this is like the forefront of medicine, so like it's really hard to say whose fault it is when you're first discovering the gene associated with the disease.
Absolutely. The gene was known, but this variant wasn't. I mean, there's thousands of letters in the gene, and only a few have been reported in the database as being disease-causing. So when you have a variant that's not been previously reported, you can only guess. Does it change the protein? Does it change the protein in a meaningful way? Those are difficult questions to ask. And this is the kind of data, obviously, that as accumulated benefits people down the road.
What was like was this actionable, this mutation?
Yeah, it was. It turns out that they could have changed treatment based on this protein being
Yes, that's
mutated.
true.
Do you think that transcriptome sequencing has the same implications as full exome sequencing if you're just looking at RNA?
Yeah, but the database is worse.
Okay.
I mean, it's more fuzzier. We have tons of transcriptome data associated with disease, but having a higher level of this particular gene isn't, is
like that.
rarely very diagnostic for what's wrong with you, because it's usually causally down the pathway, right? Something is mutated, which creates this process, which eventually creates big changes in functional metabolism. But those functional genes aren't the ones that are the original cause of the trouble, usually.
It's like having a BRCA mutation, for example. If you're just looking at a transcriptome, you may not see
You
the
won't see it.
manifestation of that at all. So.
Yeah, you won't see it because it's not a gene that's very highly expressed anyway. It's a regulator and a DNA repair mechanism. And so those are not genes that are expressed very high, and so it would be very hard to detect that.
I mean, that's
Alright.
a problem you have in a field where your like knowledge about it is literally changing every like 2-3 years. Like, you know, it's like it's just ongoing, and how much of it you can clinically apply, you know, it's tough. It's this enormous amount of data that we have. We don't know what to do with it.
Right. So in my opinion, the most critical area is building ClinVar, this association between variants and clinical outcomes. And you know, it's just slow because not everybody who's sick gets sequenced. And even if we do, we don't have really good ways of drawing the connection between the genotype and the clinical outcome,
Yeah.
what it does. So if you're interested in this sort of general area, there's this, yeah. I don't quite know why this keeps happening. It must be my computer, but
No,
No,
it's
it's
everyone.
everyone.
It's
Apples.
everyone.
Oh,
Think about
okay.
the connection.
It's funny. So somehow, right. So if you're interested in this, there's this yourgenome.org that has all these sort of ethical case studies and a quiz for you as a clinician. And they should present these things and say, okay, what should you do here? Right? And this is an example. This is a child who has a mutation with a specific cancer. This is a germline mutation that's a susceptibility to cancer. Should you share this with the child's parents? This is not what they came into the study for. It was for developmental disorders. And then one of the bullet points associated with this are, if your clinical trial protocol requires you to release all information back to every participant, you're going to be overwhelmed because there's hours of counseling associated, there's tremendous range of variability in the actionability or even the certainty of these results. And so we've seen good and bad examples in the previous two slides, right? One somewhat unknown result was hugely important in saving this person, and then this other unknown result wasn't released and it was a bad situation. But there's certainly tons of situations where if released, the data leads to confusion and stress and no actionable benefit.
Do these things get discussed up front with people in terms of, like, if we should find a mutation in something other than what this study's about, would you want us to tell you? Like, is that on the consent form, that kind of stuff?
That is part of your design of a consent form.
Okay,
So that's why we have this class, is to
That's
think
discuss
about
that
stuff
further.
like that. I mean, the easier way to do it is to say we're not going to release any unintended data back to you because we don't want to confuse and upset you.
I
But
can
maybe
imagine
not.
a study saying, like, having a blanket, you know, we'll share, you know,
All
we
data?
we find something, you know, back, because I don't think, I mean, you're not equipped, you know, to handle that.
Yeah.
And, like, you know, it takes a whole set of genetic counselors and other people, you know,
It would
come in.
overwhelm the cost of doing the study. You know, you're studying X, you're going to tell people information about X, not Y, Z, and Q.
That company is what's Illumina?
Illumina, they're the sequencing company.
Yeah, well, they have a spin-off called Grail, which is working on using just blood samples for pan cancer screening, like essentially. They're hoping